Hello. So in this lecture, we're going to basically talk about nucleic acids. So let's go ahead and write that down. Nucleic acids. So nucleic acids are the fourth type of biomolecule that we're going to be studying. Okay, so if you recall, we learned about carbs or carbohydrates, lipids, then we learned about proteins, and now we're learning about nucleic acids. Okay, so nucleic acids, there are two types of nucleic acids. Okay, DNA and RNA. Okay, DNA stands for deoxyribonucleic acid, DNA. That's where the DNA comes from. And then RNA stands for ribonucleic acid. And oops, sorry. And that's where the RNA comes from. Okay. So just like the previous biological macromolecules, these ones, carbohydrates, lipids, and proteins, nucleic acids are also pretty gigantic. Okay. See, these are huge molecules. Okay. Gigantic or in scientific terms, we call them macromolecules. Okay, so because they're macromolecules, in order to understand how they work, it makes sense to kind of take a look at, what's the simplest unit. Okay, so the simplest unit of DNA or RNA is called a nucleotide. Okay, so that's the simplest unit, nucleotide. So let's dive into exactly what nucleotides are made up of. Nucleotides are made up of three parts. Okay, they're made up of something called a nitrogenous base. They're made up of this is called a 5-carbon sugar. Now, a 5-carbon sugar is just another name for a 5-carbon monosaccharide. Okay, and then the third component is called a phosphate bridge. Okay, so excuse me. So let's take a look at some examples of nucleotides. Okay, so I'm going to go ahead and draw an example and then circle out which part is what. Okay, so take a look at this one. So let's say I have, so there's a 6-membered ring with nitrogen in it, another nitrogen out here, an oxygen, double bonded oxygen, two double bonded oxygens. There's another double bond. There's hydrogen coming off. Okay. Okay, so before we kind of dive into this part that I just drew, this is the nitrogenous base. Okay. And then this part out here that I'm drawing right now, that is your 5-carbon monosaccharide, also known as a 5-carbon sugar. Okay, so let's continue drawing this. H, OH, another H, OH. And then at the end, there's a CH2 out there, but this is connected to the phosphate group, which is also known as a phosphate bridge. Single bond. Okay, so there we have it. So that's your phosphate bridge. Okay, so when you have these three components, 1, 2, and 3, what are these three components? Nitrogenous base, 5-carbon sugar, phosphate bridge. When they come together, they form the simplest unit of a nucleic acid. So this collectively is your nucleotide. Okay, now nucleotides keep changing from, there are a few different types of nucleotides, and those nucleotides change depending upon if you switch out, you may have a different sugar out here, or you may have a different nitrogenous base out here, and when you change out the bases or the sugars, then you have a different nucleotide. Okay, so let's take a look at how this simple unit comes together in the structure of DNA or RNA. Okay, so let's say you have this unit out here, and I'm going to draw one more. Okay, so if you recall, in all the complex biological molecules, basically what you have is you have these links, right? Uh, you have these units. So let me just do it in green out here. You have these units that are connected to each other, kind of like the beads of a necklace. Okay, and whether you're looking at carbohydrates, whether you're looking at lipids, whether you're looking at proteins, those units they're connected together. And in DNA as well, these units are connected together. So in DNA, the nucleotide, so that giant unit is actually one of these, right? So now we're going to take a look at how this one connects to another one of the units, and then how that one connects to another one, and so on and so forth. Okay, so let me just go ahead and draw two of these. Okay, so I'm just going to go ahead and copy this one, this nucleotide. I'll just put it on this page, make two copies, one and two. Okay, it's kind of fun when I can just copy them without having to draw everyone from scratch. Anyway, so here we have these two copies, okay? And what I'm going to do is excuse me, I'm going to show you how they connect to each other, okay? So basically, you have the connection begins with the phosphate bridge, okay? So the phosphate bridge, so on this molecule, the bottom molecule, so that phosphate bridge, it'll connect to this oxygen out here, okay? So if I just go ahead and erase that hydrogen, okay, and I go ahead and, gonna erase that double bond, okay, from that oxygen. Well, I should probably erase this oxygen too, okay, and make that connect over to that oxygen, okay. So now what we have is, okay, we have the phosphate, and actually, in this case, actually, hold on a second. This negative charge out here gets taken away, and so essentially, you have a phosphate with five oxygens surrounding you. One, two, three. No, sorry, four oxygens. One, two, three, four. Okay, and if you start counting the carbons on the sugar, okay, so one, two, three, 4, and 5. Those are the 5 carbons on the sugar. Watch carefully if I do the same count on the nucleotide below. 1, 2, 3, 4, 5. Okay, so what's essentially happening is you're having the phosphate bridge connecting carbon 5 of the previous sugar to carbon 3 of the next sugar. Okay, so this is the key to these molecules connecting. So if I actually, kind of put another nucleotide down here, okay, I can essentially connect. Oops, sorry, wrong color. I can connect that phosphate there, put that double bond there, and then see if I number these carbons on the previous one. See, again, it's a 5-3 connection with the phosphate in between. Okay, so any nucleic acid is going to have, if I zoom out for a second, it's going to have, these nucleotide units connected to each other via these phosphate bridges. Okay, and how are the connections made? 3 on the top, the third carbon on the sugar on the top with the fifth carbon on the sugar at the bottom. the third carbon sugar on the top, the fifth carbon of the sugar at the bottom. Okay, so if you start putting these all together, now what happens is depending upon excuse me, there are the type of nucleotide. Okay, so there are a few different types of nucleotide, which is dictated by the type of a nitrogenous base. Okay, so if we go back up here, take a look at, I have my nitrogenous base, right? And I said, so it turns out, excuse me, there are four types. Actually, there are five types of nitrogenous bases. Five types of bases out here. Okay, the you have, I'm going to list them out: adenine, guanine, cytosine, and thymine. Okay, and then that's the four. These four are present in DNA, by the way. Okay, these are the ones that DNA has, four different types. And then you have another one, which is uracil. Uracil is present in RNA. Okay, and uh, now there's still going to be four. So DNA has four, RNA has four. Okay, so DNA has four, RNA also has four. But so if uracil is present in RNA, that means it's replacing one of these guys. Okay, so RNA basically has uracil, the thiamine is replacing the, uracil is replacing the thiamine in RNA. So therefore you have adenine, guanine, and cytosine. Okay, and now it's you see there's like a lot of writing out here, so we can abbreviate these. So so these are also abbreviated with capital letters A, G, C, and T. And in the case of uracil, you have a U out here for uracil. Okay, so a lot of times when you see the different nucleotides, connecting depending upon the different base, nitrogenous base structure, we typically tend to just identify them by the different base structure because the sugar is the same and the phosphate is the same. So these two don't differ. It's only this one that changes. So therefore, it's easier to kind of show the sequence based off just the different kinds of bases. Okay, so let's take a look at the sequence, an example sequence. Okay, this is also called the primary structure. Just like in proteins, primary structure was the sequence of amino acids. So primary structure of nucleic acids is the sequence of nucleotides. Okay, and here's an example: 5 prime G A T A C. Oops. C C G G. I'm just making this up, by the way. Okay, there are certain rules that need to be followed when you create a sequence because the specific sequences do specific things, but I'm just trying to show you an example of this type of sequence you can see out here. So now see what we're saying out here in the sequences, that see that 5 prime, that denotes the carbon 5 of the sugar. Okay, so it says that there you go, that's carbon 5 of the sugar, and then see it ends in carbon 3. It makes sense because 5 of one sugar is connected to 3 of another. So if you look from the top, okay, you, depending upon where you look, which side you're looking from, you would have a 5 end and a 3 end, right, depending upon where it ends. Okay, so you have the 5 and the 3 end, and then the G stands for guanine, A for adenine, T for thymine, A for adenine, cytosine, guanine, thymine, adenine. Okay, so just looking at these different types, you can clearly see that uracil is not present out here. Therefore, this is the primary structure of a strand of DNA. Why? Because there's no uracil present. Okay? So if you had a strand of RNA, okay, then there would be uracil present. So let's take an example. Let's say I have 3 prime, G, A, U, A, C, C, G, G, A, C, U, 5 prime. Watch carefully. You start with 5, you end with 3. You start with 3, you got to end with 5. Okay, they cannot end with the same number. You have to end with it's either starts with 3, ends with 5, or starts with 5, ends with 3. Okay, and then the other thing to watch out for is, notice carefully out here, there's no T, which means thymine is not there. But instead of T, you have U's. Okay, and they don't have to be in the same sequence because that's just a different strand of RNA. So this one can be identified as a strand of RNA. Strand of RNA, okay? And so now once we know the primary sequence, okay, the primary structure of your nucleic acid, let's try to understand, the concept of secondary structure. So secondary structure typically results from 2 different strands. So let's say I have one strand out here, and then I have another strand. Let's do it this way. So you have one strand out here. I'm just doing a helical structure of that one strand, and then if I have another strand, okay, I'm gonna do that in blue out here. Okay, so what happens is these 2 strands, they're connected together by hydrogen bonds, and that's what keeps them the same way. So this is the secondary structure, okay, where strands form helixes and use hydrogen bonding to retain their shape. Okay, so there you have it. Okay, now how does this hydrogen bonding take place? Okay, we can take a look at it in a little more detail. But before we do that, just so that you know, DNA exists. The secondary structure of DNA is a double helix. Okay, and that double helix looks just like this. Okay, if you compare it with RNA, the secondary structure of RNA would be a single helix, and that single helix would look like this, just a single one. Okay, all right, so let's take a look at this hydrogen bonding. Okay, so if you recall the bases that we talked about previously, okay, let's draw 2 different bases. Okay, so I'm going to draw this base. This is called guanine. I'm going to draw the base guanine. Okay, so there's the NH. There's a nitrogen out there. And there's another NH2 out there. Okay, and then this one's connected to a 5-membered ring. Sorry, nitrogen. Okay, there you go. And now this part of this part is connected to the sugar, and then the phosphate group. The phosphate, okay? And then let's say you have another base, so nitrogen. Okay, and now this one can also be connected to another sugar and a phosphate. Okay, don't worry too much about that. This base is called cytosine. Okay, so if you take a look at this, so there you have a G and a C interaction, right? G and a C. Watch, you're going to have a hydrogen bonding between that. Oh no, hydrogen bonding there between the hydrogen and oxygen. Another bond out there, and another hydrogen bond. So see, you have three hydrogen bonds. Okay, so typically between G and C, I'll just draw it out. You have between G and C, you have three hydrogen bonds, and between A and T, you have two hydrogen bonds. Okay, you don't have to be able to draw these structures out. You just need to understand that they bond. Uh, they form hydrogen bonds. Okay, and now how does this all come together? Before we get there, this kind of hydrogen bonding that we're seeing is called base pairing. Base pairing. Why? Because the nitrogenous bases, they're pairing with each other. G always pairs with C, and A always pairs with T. Okay, so they're also called complementary nucleobases. Okay. Okay. So basically, G tends to combine with C, and A tends to hydrogen bond with T, and that's pretty much what complementary nucleobases are. Okay, so how does this all come together in that double helix of DNA that we talked about? I'm going to take a picture of this, and I'm going to just show you that picture so you can kind of see how it comes together. We'll discuss that a little bit. Give me a second. All right. Let me pull that picture down here. Let's take it to a new page and just zoom in here. Okay, let me crop this so we can focus on. All right. So now you should be able to see. This is just a very rough schematic of DNA. The two strands of DNA that come together. Notice carefully the complementary base pairing A and T, G and C, C and G, A and T again. And then watch carefully how you have the primary sequence. Okay, the primary sequence. This strand is going starts off at the 3 end, and that's the 5 end. And then you have, this would be 3 T C A C G T, and then ending with 5. And on the other end, you have this complementary strand, which is going to be 3 A C G T G A, ending with 5. Okay. And that's pretty much what you need to know for this topic. Um, if you have any questions, feel free to let me know. Otherwise, have a good one. Thank you very much for watching. Bye.